So this lecture is particularly on the topic early diagnosis of human genetic diseases. Now, many diseases have a genetic component. However, the extent to which the genes contribute to disease varies and much to be learned. Advances in understanding various genetic, the mechanisms behind these diseases help to devise early diagnostic tests. Now, early detection of genetic diseases in newborn or the individuals yet to be born may help in, early intervention and treatment, which may either cure the disease if it is a metabolic disorder, or improve the quality of the life the individual is going to lead. And many times it leads to prevent the disease, or intervention is practiced to minimize the severity of the disease. Now, detection technology for genetic disorders, there are many challenges. The population genetic variations, there are lots of polymorphism, and so methodology of genetic screening of a population makes it a very challenging job. But there are some, reliable, the prenatal diagnosis and safe methodologies there, which we'll talk about. And there are different tests of genetic disorders like carrier screening. Those who are heterozygotes, they should be screened. And pre-implantation genetic diagnosis, prenatal diagnostic test, newborn screening, pre-symptomatic testing for predicting adult onset disorders. So many of the disorders which cannot predict beforehand, and then it manifests later in the age. So first start off with the prenatal genetic the diagnosis, and before that a few words on the quad screen test. What do you mean by that? The quad screen is also called maternal serum screen. Is a prenatal screening test that analyzes 4 substances in your blood. In Latin, the quad means 4. It's usually carried out between the 15th to 22nd week of pregnancy. The quad screen tests 4 factors in your blood to help you make a decision about further genetic testing. It's a blood test that measures the level of 4 substances produced by the fetus and the placenta, and one can find out in the mother's blood serum. One is the alpha fetoprotein, a protein which is made by the fetus, human chorionic gonadotropin, hormone released by the placenta, then estriol, a form of estrogen produced by the placenta and fetus, and inhibin A, a hormone made by the placenta. So the 4 important, substances are tested, and together they are entered into a formula that tells you what are the probabilities that you are carrying a child with a chromosomal abnormality or not. Quad screen, one of the important parameters like neural tube defects can be detected. High levels of alpha fetoprotein, AFP, may suggest the possibility that a baby is at higher risk of neural tube defects, including spina bifida. And chromosomal abnormalities are there. Low levels of AFP and abnormal levels of the other markers may indicate that the developing baby may be at higher risk for chromosomal anomalies. Okay, you can see the neural tube defect. You can see the baby, the lower panel, the upper panel shows the ultrasonogram. Then on the backbone, if you look at where the spinal cord is, the inside, you can see there's a protrusion. Okay, so this is a spina bifida. Because of this gap, and the spinal cord comes out. Okay, and although they survive with lots of protection and all that, but this is can be prevented if that can be known beforehand. And so what happens? The alpha fetoprotein keeps on leaking out so their level becomes abnormally high. So like all prenatal screening, the quad screen does not provide a conclusive answer or to whether or not your child has a genetic abnormality. It can only tell you whether your risk is more or less than the typical risk at your age. When this test should be done, it's typically recommended for women who are already in the second trimester between week 15 and week 22 of pregnancy. So this is a calendar, and different genetic disorders, the early prediction can be done sometimes in this early second trimester, or it can be the first trimester. The amniocentesis, very popular and common the prenatal diagnostic methods. Specific reasons why an amniocentesis may be recommended: because the first trimester or second trimester ultrasound shows something wrong, or abnormal results on peripheral blood screening tests such as quad screen shows something they like to worry about. Family history of certain genetic conditions, you or your partner, this shows that there can be a genetic disorder, okay, in the future generation. And maternal age of 35 or older, the risk of chromosomal abnormalities increases significantly with age, especially after the age of 35. In amniocentesis, a hollow needle is inserted through the mother's abdomen into the uterus, and amniotic fluid is drawn for analysis. This is how you can see the outermost covering is the chorion, and then comes the amnion, and the cavity is called amniotic cavity. And there are many fetal cells, the plausa from the fetus, and you can collect those cells. Although your insertion of the syringe should be guided by ultrasonogram camera. Okay, that is very, very important. Otherwise, there is a risk that you can injure the fetus. And so always, it is done through a ultrasonogram, there's a ultrasound transducers with a technological guide. So basically, when you see the ultrasonogram, ultrasonography, the aided such type of, the collection of the samples, and then, you can collect maybe 30 ml of amniotic fluid, and then, you can, this is how you can examine under microscopes, and then, you can centrifuge it, and then you can pellet it, and you can do the or rest of the procedures like these adding of hypertonic solution, fixations, and then they make chromosome preparations and so on. And then also you can, from the fetal cell, you can also do the molecular biology based lots of testing, and you can do DNA, the analysis. You can do PCR testing after they isolate the DNA. You can do biochemical analysis to look for the metabolic disorders and so on. The other technique, which is Boronicvilli Sampling, it can be done at 8th week and up to the 10th week of gestation. And the cells obtained from the Boronicvilli is very, very prolific and can be cultured easily, in contrast with amniocentesis, where cells are not so prolific in culture and procedures cannot be done before 12 to 16 week of pregnancy with some effect on the mother. And you can see that the chorionic sac, amniotic sac, and so chorionic sac, it's very, very prolific cells, and you don't have to go deeper. Like for amniocentesis, you have to collect samples from the amniotic cavity. So here you can collect your samples from the chorion. And similarly, you need the transcervical like sampling and with ultrasound transducer. If you compare the amniocentesis and CVS, so amniocentesis is a small sample of the amniotic fluid surrounding the fetus is removed and examined, and living cells from the fetus float in the amniotic fluid, usually done in the 14 to 16 weeks into gestation. On the other hand, the Boronicvilli sampling, tiny tissue sample from outside the sac where the fetus develops, and this tissue put into the culture, and tests are done for chromosomal DNA and other tests. CBS can be performed as early as 8 to 10 weeks of gestation. Now, if you look at the period of sampling, now for amniosynthesis, 14 to 16 week. Boronicvilli sampling you can do much earlier. So many people, they prefer, the Boronicvilli sampling, that you can know much before the pregnancy, the proceeds further. And if there is a genetic, the disorder, they predicted, then one can go for the medical termination of pregnancy. So you can do karyotyping, biochemical test, and so on. Now, coming to the last part, that is a prenatal and pre-implantation diagnostic methods of genetic disorder. Before the fetus, they started, they developing, right at the implantation stage, can we do the, the disorders testing? So let me discuss an associated factor called general causes of infertility. So there are male factor, there are female factor, but there are unexplained factors also. So many of these such, the couples, they go for like to IVF clinic, in vitro fertilization clinic. Now there are genetic causes also. It can lead to infertility, like genetic abnormality, chromosome abnormality, abnormal embryos. Then there is an implantation failure. So why for such, the cases, you go for in vitro fertilization. So it can be option if a woman's fallopian tubes are missing or blocked, a man has low sperm counts, unexplained infertility has continued for a long time, and a couple wants to test for inherited disorders before embryos are transferred. Okay, so this is what we'll be discussing. And so this is a overview of the in vitro fertilization, since it is in the practice since 1979. The conventionally you can see that the when the zygote divides 1 to 2, 2 to 4, 4 to say 4 to 8. Then you can see that at certain stage you can remove the one of the blastomere, and you can do the testing, karyotype testings, or you can do the DNA testing and so on. Now, if you test result shows that it is genetically abnormal, so you can accept or reject, and you do not go further to implant. So so pre-implantations diagnosis occurs at this stage. So you can take a decision, accept or reject. And these are other the advantages of are there, where you can keep the frozen it's like the later on that can be used for the future pregnancy. One can make tissue factory if the individual after he or she is born, there are some kind of injury. So those tissues can be used for the treatment. So it cannot be the self tissue when it is implanted to the own body. So there will be less immunological hazards. And about the artificial womb and artificial chromosome, it sounds like a fiction now, but in the future, you may not know that there can be such type of things happening, and this may sound fiction right now. Pre-implantation genetic diagnosis, this process of screening embryos for genetic abnormalities prior to transferring to the uterus. It requires IVF, and this refers to procedure that perform on embryo prior to implantations. And you need a micromanipulator and inverted microscope as a platform, and you can see the blastomeres. After the zygote formation, quickly they divide. So you can take a small, side blastomere, and then you can do the analysis. So here you can see some of the videos, how it is done. So this is done under microscope. And one of the important things to notice that day of egg removal, this is the calendar, day 1, day 2, day 3. So from day 3 to day 5, you can take out a single cell, and then you can carry out DNA testing. You can undertake chromosomal the analysis and so on. So this is a sort of summary slide that the egg and sperm, when you fuse them, fertilize outside the body, we will call it in vitro fertilization. Remove one cell on the day third and test the DNA chromosome, and the test results, if it's a healthy, then a condition embryo is implanted on the day 4. If it's unhealthy, it's discarded. Who should consider PGD? For a carriers of single gene disorder or a carrier of sex-linked genetic disorder, have a history of recurrent pregnancy loss. In some instances, PGD can also be used in the gender selection process. See the video. So thank you all. I hope these informations will be useful to you. Thank you.